Yes, thank you very much. Obviously, thank you to the organizers for the opportunity to be here today and share some of the work that we've been doing over the last several years. I think it's been a really great meeting. And what I want to talk to you about today, I think, is something that we all think about very deeply, and that is how are these tumors developing in the context of what's a very complex ecosystem of cells. This is just depicting sort of early tumor evolution as we all sort of appreciate that these tumors can undergo early immune surveillance, which may select for clones that are more competent to escape that immune response and grow out. And I think oftentimes we think about this very much as an interplay between the tumor cells and their surrounding immune hematopoietic environment, and how that sort of goes on. And what I sort of suggest, and again, I don't think I have to harp on this too much, is that the adaptations and the maladaptations of the underlying tissue structure is also very critical for determining these very early tumor evolutionary processes. And I think if we can understand sort of that context within which these tumors are evolving, provides us really keen insights into how tumors progress, and maybe some of the fundamental things that lead to the phenotypes that we see when these tumors present clinically. And so the structural component that we think a lot about are the lymphatic vessels. These are the sort of requisite vessels that direct, obviously, fluid homeostasis, so the movement of interstitial fluids out of our tissues to be filtered through lymph nodes. They play a very important role in biasing local morphogen gradients. They present and express the chemokines that are necessary for migratory dendritic cells to make their way to the lymph nodes. And very nice recent work has also shown that they can produce factors that support cells within really novel niches. And so I think we're gaining a really broad appreciation for how dynamic and interesting this system can really be, and the important role that it's playing both at tissue homeostasis and in the response to inflammation. And you can see these vessels here. These are these larger caliper green vessels that we stain for live one in the mouse skin. Now, Cyrus alluded to this in the context of cancer. I think for a long time we had primarily thought about the lymphatic system as a passive conduit for metastasis. Many solid tumors present early with lymph node metastasis, and this is generally a poor prognostic, though not in all cases. But as Cyrus really nicely described to you, complete lymph node dissection has actually not moved the needle for patients with respect to decreased rates of recurrence. And so we've really truly don't understand, one, why the lymph node is such an early spot for metastasis, or what the impact of lymph node metastasis is on the systemic host. We'll come back to that idea, but the thing that we wanted to challenge when I started my postdoc was the idea that that's all they did, that they simply, by being a conduit through which things can move, if you have that conduit proximal to a tumor, then tumor cells can move, and therefore things are bad. And the reason we wanted to challenge that idea is because there's something else lymphatic vessels are very important for, and that's for immune surveillance, again, both at steady state and in response to inflammation. And if you look at sort of the classical tumor immunity life cycle as presented by Ira Melman, the lymphatic transport governs an entire half of this life cycle, again, being the really requisite route for the presentation of antigen in lymph nodes, and then the redistribution of that effector response from the lymph node back to sites of inflammation where they exert their effector function. And so we really thought that there was sort of a lack and sort of a gap in our understanding for how the system must be contributing to tumor progression, and really presents this interesting idea, which we'll talk about today, is how can these vessels be at once bad in the sense that they allow for dissemination, but also crucial in the sense that they drive these immune responses, and can we think about ever decoupling these processes? So one of the central hypotheses in my lab is that these are not passive structures, these are active structures, that the endothelial network itself can respond to contextual cues, and that can actually lead to qualitative and quantitative differences in immune responses. That's really critical for just how our system is adapting in the context of immune challenge. And I'm going to show you very quickly some data that we generated in viral models, which have really solidified this idea for me. And what we did was we took advantage of a skin infection model. This is a vaccinia virus, which we deliver by scarification. And one of the first observations that my lab made, actually, was when we do this, rather than seeing more fluid flow out of that infected tissue, which is what the textbook would tell you happens, we saw a rapid decrease in fluid flow out of the tissue. And that decrease in fluid flow associated with this remodeling of the lymphatic capillary from what we describe as a very open, button-like structure in the naive setting to this much more constricted vessel with continuous interendothelial junctions. And this actually depended upon VEGF-A and VEGFR2, so I enjoy the non-vascular mechanisms of bevacizumab. So what does this mean functionally? Well, we actually showed that when these vessels actively respond to VEGF to constrict their junctions and prevent fluid uptake, that this is actually optimizing the response to the virus. So such that if we inhibit VEGFR2 signaling specific in the lymphatic endothelium, we rescue fluid flow out of this tissue. This now actually allows virus to disseminate, so the lymphatics are acting as an innate barrier to pathogen spread. But perhaps more importantly and excitingly, this release of fluid flux out of the tissue actually decreases migratory dendritic cell migration and thereby decreases the probability of CD8 T cell priming. And so in this way, this sort of paradoxical tightening of the lymphatics is somehow optimizing the compartmentalization of antigen for improved responses. And in unpublished work, we actually see that this has significant impact on germinal center responses as well. And so again, this told me that we have to be a bit more nuanced in how we think about the lymphatic system with respect to how it might be regulating the responses to peripherally restricted antigens, much as it is in tumors. And so if we come back to tumor, then how do we translate this knowledge, right? So in the context of cancer, we of course know lymphatic vessels are still required for tumor immune surveillance. Many of the mechanisms used during viral infection are shared, so dendritic cells migrate along CCL21 gradients that are generated by the lymphatic vasculature. And we see in many tumors an increase in expression of these lymphangiogenic growth factors, VEGF-C and D, which drive the expansion of these vessels, and that's often associated with increased lymphatic flow and increased dendritic cell migration. And so it has been presented in the literature from several different groups that perhaps one strategy to improve tumor immune surveillance would be to increase flux through this pathway, leading to more antigen presentation in the lymph node and more effector responses, and this would be overall good. But I just showed you that in the setting where we give beautiful antiviral immunity and long-term memory, the exact opposite of that is happening. And the other problem with that hypothesis is that you're going to always get more metastasis in this setting, and this, we know itself, can drive more immune suppression. So there's something about this that we don't fully understand, and we have to sort of think more deeply, I think, about lymphatic biology. The other thing that kind of counters this idea of just more lymphatic vessels should inherently be better is if you look in tumors, the genetically engineered tumors, for example, this is an inducible melanoma that is induced in the skin of these mice, and you can see it forming here in the dermis at late stages. These tumors have beautiful networks of lymphatic vessels that have greatly expanded over time, and absolutely no CD8 T cell response. So it can't be possible that lymphatic vessels themselves are sufficient to boost tumor immune surveillance. So how do we think about this problem a little bit differently? And so a graduate student in the lab, very talented, Filio Karakusi, was really interested in this problem, and we were sort of trying to sort of think about how we come at this in a different way. Almost all of the work that had been done by us and others had taken the approach of overexpressing growth factors to drive growth and to ask how that perturbed the system. And we wanted to think about doing this in a slightly different way because, as we just heard, these growth factors can affect many different types of cells within the system. So Filio decided to take advantage of a system and sort of ask this question in reverse. So instead of boosting the lymphatics and ask how that impacts immunity, let's change the immunogenicity of the system and ask how that impacts the lymphatics. And to do that, we take advantage of two melanoma lines generated by Marcus Bosenberg at Yale, the YUM and its daughter line, YUMR 1.7, that differs because it's been exposed to UVR radiation and so has an increased burden of somatic mutations that render it more immunogenic and responsive to checkpoint blockade. And what we simply did was look at the lymphatic vessels, which you can see here in red, within the tumor itself. And what we saw was sort of surprising given the literature context that we had, which is that in both cases, you can see the pre-existing lymphatic vessels here in the normal dermis adjacent to the tumor. But really only in this, what we would term a cold tumor microenvironment, are you seeing the extension of the lymphatic vessels down into the parenchyma of the tumor itself, whereas they are largely excluded from the more immunogenic tumor. So this already starts to hint at this idea that it can't be, again, that just more vessels are at least required to get really potent immune responses. And this aligned really nicely with single-cell RNA-seq data generated by Chris Marine at the VIB in Leuven, where he had actually found that when you look at patients that are responding to checkpoint relative to those that are not responding, and you look at the abundance of lymphatic endothelial cells collected in these specimens, that responders had, by and large, far fewer lymphatic endothelial cells, and that number further went down on response, on treatment, relative to baseline, with no change in non-responders. And if you just look at the relationship between infiltrating immunity and lymphatic endothelial cells, you can see this really mutual exclusivity, which suggested to us that there's some crosstalk between these components that might be really interesting for us to explore. Now, these results, which implicated interferon gamma, as well as the work we had been doing in viral infection, which showed this beautiful activation of the lymphatics in the presence of a high interferon environment, got us interested in asking whether interferon could be playing a direct role in regulating this process. And so we took advantage of a mouse model that we had in the lab. This is an interferon gamma receptor depletion specifically in the lymphatic endothelial cells using a PROX1 CRE, which is specific for the lymphatics in the skin. And what we saw was really, really exciting to us is we take that immunogenic tumor, we implant them into wild-type mice. We see what I showed you before, very few lymphatics within the center of the tumor. But by simply making the lymphatics sort of blind to the interferon gamma, we could enhance and sort of rescue this intratumoral lymphangiogenesis. Now, at this time, we were interested in understanding how this impacts metastasis, and these YUMR tumors are not very metastatic in our hands. And so we decided to turn to a model that we had developed where we overexpress VEGF-C. And I thought this would actually not work. I thought that the sort of superphysiological levels of growth factor would wash out any kind of endogenous regulation of the system. And that was just completely wrong. Because even in the presence of this super high levels of VEGF-C, which do increase the intratumoral lymphangiogenesis that you can see here. When you release the lymphatics from the negative stimulation of interferon gamma, you just see this tremendous expansion of vessels into the tumor itself. Does this impact metastasis? Yes, it does. So you can see metastasis to the draining lymph node here indicated by pigmentation is significantly increased in the absence of interferon gamma sensing by the lymphatics. And I think this actually aligns really nicely with some of what Cyrus was saying as well. So this, to me, was really all very expected. The bigger question that I presented in the beginning is it ever possible that you could have an expansion of the lymphatic vasculature that was pro-metastatic but not productive in this context of an immune response? And so we now took this model and we asked whether or not the sort of initial steps of immune surveillance would benefit from this significant expansion of exit routes from the tumor, if you will. And the answer was really surprisingly no. Okay, so we can look for, in this case, CD11C positive cells within the tumor. We can, of course, find them there. They're proximal to these intratumoral vessels, both in the wild type and in these knockout mice. But if we use, in this case, FITC paint to actually track the migration of these DCs from the tumor to the lymph node, what you find is a significant decrease in the proportion of dendritic cells that are making their way out of these tumors, almost as if they're sort of stuck in this sort of expanded lymphatic plexus. And consistent with this, there's no benefit for immune checkpoint blockade. So that sort of small benefit that we got with the overexpression of VEGF-C is not further enhanced by this expanded network. So for us, this was really exciting, first proof of principle that these processes could be decoupled, and it suggests that if we understand the underlying biology here, that we might be able to take advantage of this from a therapeutic perspective. And so the questions that this raised for us was that are all lymphatic vessels not then created equal? Could there be activation states, much as I'm sure all of you think of your cell populations as having activated and quiescent states? Could lymphatic vessels similarly adapt to their context in this way? And can we define those and then use those states actually to tune these various processes that we know are being co-opted and used in the context of the tumor? So to get at this, the first thing that Filio did was she performed single-cell RNA sequencing on these tumors, and I think there were really two predictions. In support of this hypothesis, we might see the outgrowth of a novel transcriptional state that would suggest that there's something really truly different about these vessels in the absence of interferon gamma, or sort of disproving the hypothesis would be that we simply just expand equally all different lymphatic clusters, if you will. And so probably would not be a very exciting story if the hypothesis didn't bear out. But we were really excited to see this data, because when you look at lymphatic endothelial cells, again, these are lymphangiogenic, right? They're growing in the context of VEGF-C. We have these 4 different clusters, and when you get rid of the interferon gamma, we do not see equal expansion of all of these clusters. What we see is a very specific expansion of this cluster 3 here shown in red. And so this was the first inkling that there's really something different about these lymphatic endothelial cells. So what's different? We can perform pseudotime, and we can demonstrate that these are likely pre-existing lymphatic endothelial cells that progress over to this sort of novel cluster 3. And if we start to map different genes within this pseudotime space to ask what is really what is this new state that these cells are entering, what you can see very clearly is that first there's this proliferative bottleneck here in cluster 2 that's activated, and then as these cells progress into this cluster 3, what they really look like they're doing is entering this very developmental, invasive, pro-proliferative program with high expression of transcription factors that are absolutely required for lymphatic development. And so sort of to us began to suggest almost not a true de-differentiation of the cell, but perhaps a return to a more immature developmental state. What was quite exciting is that we could take a look. This is a data set that was recently generated, published in Nature, pan sort of vasculature atlas from tumors with a really nice population of lymphatic endothelial cells. These are actually difficult cells to represent in your data, but you can see that they have about 9,000 lymphatic endothelial cells taken from across different solid tumors, and we're able to construct a trajectory of differentiation starting from normal lymphatic endothelial cells taken from normal tissue, and what they predicted were 2 lineages. One lineage that moves towards what they termed an antigen-presenting lymphatic endothelial cell with high expression of MHC molecules, et cetera, and a second trajectory which they associated with lymphangiogenesis that came down over here. And so we wanted to ask whether or not some of the things that we're seeing with this developmental program would bifurcate in the way that we would expect. And the answer is yes. So the first thing that we were able to see is that this sort of interferon gamma program, which we were manipulating functionally in our animals, was actually defining this first trajectory here towards this antigen-presenting lymphatic endothelial cell, which I think you would expect. And consistent with our data that interferon gamma is sort of loss of interferon gamma signaling would drive lymphangiogenesis. These same transcription factors, which we found to be elevated in the absence of interferon gamma, are truly defining the move on this trajectory towards what they term a tip-like state, a light state, and we agree with, so I'll show you that data. So to me, this is really exciting, suggesting we now have a mouse model that's actually modeling the normal differentiation trajectory of what we do see truly in patients. So what do these cells look like? That tip-like state that they alluded to is exactly what we see. These endothelial cells have really high expression of Notch receptors and Notch ligands, things that are typically expressed in the angiogenic vasculature on these tip cells that get specified very specifically to allow for the proper sprouting of the endothelium. And when you have too many of these cells, you get exactly what we showed you. You get this really nasty plexus, disorganized, sort of frustrated endothelial growth, and that's what we see. I won't talk about this a lot more, but the other interesting facet of these cells is that they produce CCL2. They become in some ways very sort of immune interacting, and this is something we're very excited about thinking about even more as we move forward. We've used DLL4 as one of our markers for this network, and we can show that it's really those intratumoral lymphatic vessels that start to express this marker, and the pre-existing vessels that are out in the skin sort of persist in the absence of these. And so we really think it's these vessels that are represented by our cluster. And we think that some of these surface markers now become a unique and novel scaffold that tumor cells, but not dendritic cells, can take advantage of. And that's because if we do a transwell assay with DLL4 as an example, DLL4 itself is sufficient to boost the migration of human melanoma cells, and this has also been shown by others, and has no impact at all on dendritic cells. And so there's something that these tumor cells can really take advantage of here that does not benefit our normal mechanisms of host surveillance. And again, just to show you that data again, these programs we also see bifurcating that mutually exclusive program that was found in humans. So to finish here, I just want to show you what we think is actually regulating this lymphatic fate and growth transition in adult tissues, because I think that's obviously what's going to take us to thinking more about the therapeutic possibilities of understanding this. And so we knew from work from Guillermo Oliver, who's at Northwestern, who studies developmental lymphangiogenesis, that mitochondrial respiration is really important for providing positive feedback on this PROX1 transcription factor, which enhances sensitivity to growth factors, and what he said sort of maintained lymphatic endothelial fate in the developmental sense. So it's absolutely required for these cells to sprout out the cardinal vein and form the plexus. And so we were curious if there was something similar going on here that would be downstream of interferon gamma. And that's really what we saw. I'm going to kind of move through some mechanistic data a little bit quickly, but I'd be happy to talk about this more. But first, of course, we can see that our lymphatic endothelial cells are intrinsically more proliferative within these microenvironments when they're insensitive to gamma. If we look at that cluster 3 and compare the differentially expressed genes between wild type and interferon gamma deficient lymphatics, you can see many pathways come up, including the Notch pathway, but also things like oxidative phosphorylation and reactive oxygen species, perhaps suggesting that these cells, in the absence of interferon gamma, have kickstarted OXPHOS and are thereby re-entering this developmental state that Guillermo described. Indeed, if you look at mitochondrial ROS as a surrogate for OXPHOS on cells taken directly ex vivo, you can see elevated mitochondrial ROS only in these lymphatic endothelial cells that are not sensing gamma. Gamma itself is sufficient to do this in vitro, which is what I'm showing you here. gamma suppresses oxidative phosphorylation and mitochondrial ROS. So can we connect these things? Is it possible that a suppression of OXPHOS is itself causal, both for the proliferative phenotype as well as for the acquisition of this novel state? And we think the answer is yes. If we look at the transcriptomic analysis of human lymphatic endothelial cells treated with interferon gamma or treated with antimycin A, which suppresses complex 3 of oxidative phosphorylation, mitochondrial respiration, what you see is both of these perturbations are sufficient to really drastically downregulate a core program of genes that we associate with this TIP-like state. And to really ask if these are acting in concert and that interferon gamma is acting through complex 3 in order to mediate these responses. We took advantage of a bacterial enzyme, this alternative oxidase, which is sufficient when expressed in cells to bypass complex 3 and allow for continued electron flux through the mitochondrial respiratory chain, but doesn't generate the same reactive oxygen species, which is important. And what we find is that AOX is sufficient to rescue the proliferation of our human lymphatic endothelial cells, but insufficient to rescue these transcriptional changes. And so if we put this all together, what we really think is happening is that interferon gamma is a maturation signal. It keeps adult lymphatic endothelial cells in a quiescent, mature state. And loss of interferon gamma signaling allows for reactivation of OXPHOS, generates lots of mitochondrial ROS, and these two things act in consort to drive both the proliferation and this sort of novel state. Okay, so I just want to come back quickly to human and to what we think this means for our patients. We can see, we sort of can go back to that immunotherapy trial that Chris Marine led and actually see that lymphatic endothelial cells in responder patients who have more gamma are totally shut off the program that we see in our cells, as well as programs of just sort of notch and tip signaling that we can derive from the literature. If we stratify the melanoma cohort in TCGA, specifically by signatures that define lymphatic endothelial cells together with these TIP-associated genes, we are able to stratify outcome. And this does not work if we use genes that would be associated with like a stalk or more quiescent lymphatic. And so I think this is really some of the first data to suggest that it's not a quantity thing here, it's really a quality thing. And I think we have some insights to how we deal with this. And I'll just say that this Nature paper also associated that very lymphangiogenic cluster that they found with poor overall survival in HCC. Okay, so with that, I just sort of want to summarize. We, I think, have demonstrated that interferon gamma protects against regional dissemination through a lymphatic intrinsic mechanism, which is, of course, on top of the many other things that gamma and CD8 T cells are doing. That it's doing this by really restraining this immature state, and that we see this in patients as well. And if I put this all together for you, what this is beginning to really make us think is that lymphangiogenesis, which we see in tumors as well as in chronic inflammation, may actually be the result of failed upfront immunity. Because productive priming, productive cytotoxic immune responses, I think can keep us in this early part of this loop, and this is going to be beneficial for our patients. And it's only perhaps in the absence of robust neoantigens or active mechanisms of immune suppression that intratumoral response fails, and we get pushed over here, we drive lymph node metastasis, and we drive progression. And so with that, I will end. Filio did the vast majority of the work that I presented to you today. Thank you for your attention, and I'm excited to hear your questions.